Good morning, ladies and gentlemen. My name is Alicja Staśczak, and it is my great pleasure to present our research project on the application of microfluidic technologies in biomedicine. This project is a part of a broader initiative at the Biotechnology Center of the Silesian University of Technology, which brings together an interdisciplinary team: biologists, chemists, engineers, and programmers. We focus on using microfluidic systems, also known as lab-on-the-chip technologies, to simulate cellular environments and enable highly controlled biological experiments. This system allows for manipulating tiny volume of fluids and can precisely control fluid flow, concentration gradients, and cell behavior. This approach offers a powerful alternative to traditional petri dish and flask-based experiments, which are not only time-consuming but often fail to replicate the natural biological environment. Microfluidics provides a more realistic, faster and cheaper method for studying cells, drug interactions, and disease processes. Okay, so why is microfluidics such a game changer? First, it allows for miniaturization, which dramatically reducing reagent use and cost. Second, microfluidic devices provide precise control over experimental conditions, something that's difficult to achieve in a microscale system. In addition, cells behave differently in the microenvironment. In microfluidic system, we can simulate in vivo-like conditions such as gradients of nutrients or oxygen, flow-induced shear stress, and another condition. This enables more physiologically relevant experiments and more accurate predictions of how cells or tissue will behave in the human body. Let me now walk you through the structure of our example of microfluidic chip. It consists of three key layers. The bottom layer contains incubation chambers and microchannels. This is where we culture the cells. It includes input-output ports and degassing channels to remove air bubbles. The middle layer is a porous membrane. It allows molecules like nutrients or drugs to diffuse through while preventing direct contact between compartments. The top layer includes fluiding channels that allow us to introduce cells, reagents, or stimuli. This configuration allows for co-culturing multiple cell types, simulating communication between tissues, or creating barrier models, like the blood-brain barrier or the intestinal epithelium. By modifying the membrane of the channel geometry, we can study phenomena like cell migration, hemotaxis, or drug prevention. The microfluidic platform was designed to best replicate the characteristics of the in vivo microenvironment, which still remains a challenge. In response to this issue, a system concept was developed and modified in such a way as to enable effective and highly efficient delivery of selected drugs to the reaction chambers. The microchamber device, combined with a matrix based on glycol microgels, was designed to allow for long-term microscopic observation and analysis of cell population growth under in vivo mimicking conditions. Mostly commonly used long-term cell culture techniques required periodic medium replacement. Moreover, medium exchange leads to a sudden change in the composition of the environment surrounding the cells, exposing them to shock. This variability excludes the possibility of long-term studies of cells under stable condition. The continuous microfluidic approach applied in the device allows us to overcome this issue. A second advantage over standard cell cultures stems from the geometry of the microchambers in the presented system and the option to simultaneously deliver several substances or their variants at once. The development and commercialization of such a microdevice will enable the creation of a functional and reproducible models of cell development, which will reduce the need of animal-based clinical research. The product, together with dedicated software, will allow for the adjustment of individual doses for specific patients, making medicine more personalized and efficient. Microfluidic models require a small number of cells, which make them suitable for samples of limited size, such as biopsies. Moreover, they may have a greater potential to predict treatment response compared to the traditional assays. To further mimic the native tissue environment, we use a thermosensitive hydrogel as a 3D cell culture matrix. This material behaves like the extracellular matrix found in human tissues. It provides mechanical support, enables nutrient diffusion, and allows cells to grow in 3D structures. What makes our hydrogel special is its temperature sensitivity. It's liquid at the room temperature, making it easy to inject into microchambers, and it solidifies at the body temperature. This way, we can load cells in suspension and let them form colonies after gelation without mechanical stress or chemical cross-linking. This system is biocompatible, tunable, and highly reproducible, making it ideal for long-term experiments, cancer research, stem cell studies, or drug screening. The image on the slide shows how cell grows in the hydrogel. Unlike 2D cultures, where cells spread flat on the surface, in 3D they form natural-like spheroids and colonies, closely resembling tissue architecture. This enables more accurate studies of cell proliferation, signaling, and drug response. For example, cancer cells grow in 3D often show higher resistance to chemotherapy, which is consistent with clinical observations. So, using this model allows us to better predict treatment outcomes and optimize therapies. Based on previously developed patented solution, 2D designs of the microdevice are prepared using AutoCAD software. Geometrical solutions are proposed to ensure the highest quality of mixing and optimal drug testing conditions. Various configurations are designed for connecting the main channel with the drug inlet, as well as for the arrangement of the cell culture chambers. The use of AutoCAD enables the preparation of the tight microdevice layouts and allows for rapid correction and refinement of designs. The completed 2D layouts are then implemented in ANSYS Fluent software where 3D geometries are generated. The combination of AutoCAD for microfluidic design and ANSYS Fluent for virtual testing enables a quick evaluation of drug diffusion quality with the medium and accelerates the selection of the optimal solution. The finalized AutoCAD design is used in the fabrication of the actual microdevice. Once the AutoCAD design is completed, a corresponding physical microchip is manufactured. For previous research and development work, a specialized protocol was established to ensure the professional and precise fabrication of microchips. An important stage in this process involves the development of inlet ports to ensure their thickness and prevent leakage. In the proposed project, a numerical model of the diffusion process of the tested drug in the medium is used to visually test the designed microfluidic layouts before introducing them into production. The computer simulation makes it possible to precisely assess whether the diffusion of the drug from the medium flowing through the main channels ensures uniform mixing of the substance. This is essential to guarantee identical testing conditions in each cell culture chamber. Running simulations allows for rapid testing of different geometrics without the need of manufacture each version. This approach saves production time, reduces manufacturing cost by limiting the number of fabricated chips. Creating microfluidic chips requires high resolution and rapid prototyping tools. Traditional photolithography is accurate but expensive and time-consuming. That's why we focus on two faster, more affordable approaches: laser ablation and 3D printing. We use a high-precision laser system combined with a motorized microscope stage. This setup allows us to observe and engrave microstructures at the same time, reducing alignment errors and fabrication time. The resolution is below 40 micrometers, which is sufficient for biological experiments. Additionally, we develop custom software to control the laser and stage, allowing us to import 3D models, adjust laser parameters, and monitor fabrication in real time. The second approach we use is resin-based 3D printing to produce molds for PDMS casting and microfluidic chips. This method allows us to quickly test different designs, geometrics, and channel layouts. We tested several resins and found that TR250LV works best. It's biocompatible and gives sharp, detailed prints. Our optimized mold design includes, for example, 15 culture chambers, each with fluidic connection, air trap, and degassing channels. We fine-tuned print settings such as exposure time and base layers and added post-processing steps: UV curing, alcohol washing, and thermal stabilization. A common problem with 3D printed molds is that residual monomers or photoinitiators inhibit PDMS curing. To prevent this, we use extended UV curing and heat treatment, followed by surface siliconization, which forms a protective barrier. This ensures that the PDMS fully cures and doesn't stick to the mold. Here we have example of microfluidic platform combined with thermogel matrix based on glycomicrogels. Once the chip is fabricated, we load the hydrogel and cells and run the experiments. We can introduce chemical compounds, control flow rates, and monitor cell response over time using live cell imaging. The data we collect includes cell viability, morphology, migration pattern, and drug response profiles. This information is valuable for preclinical drug testing, understanding disease progression, or studying interactions between different cell types. Here we have example of time-lapse microscopy images of culture microchambers. After that, we are doing data analysis. Based on the conducted analysis, the obtained information provides a solid foundation for understanding the reactions that have occurred in the system, taking into account the limitations specific to the applied technology. An extremely useful and essential aspect is the comparison of the analysis results with the developed numerical model of the diffusion process of the tested substance in the medium based on a microfluidic system, which constitutes one of the stages of the project. As part of the work, microscope images acquired at strictly defined time intervals are taken into account, serving as a valuable source of information. The conducted analysis allows for real-time assessment of the development of human or animal cells and the process occurring within them. Microscopy is mainly used to study the dynamics of immobilized cell growth under condition of continuous medium exchange dominated by diffusion, monitoring not only cell development but also their biological equilibrium. Image data analysis mainly involves cell segmentation and counting. After counting, the cells undergo quantitative evaluation based on a series of images at each time point. Morphological filters are also applied, which facilitate operation related to the size, shape, and consistency of the cells. The use of these filters enables the elimination of potential artifacts. Since the accuracy of morphological measurements largely depends on the image processing and segmentation, an extensive analysis is carried out to optimize the applied techniques, including linear and non-linear filters, edge detection, and morphological filters for binary and grayscale images. As part of the work, a user-friendly application with a graphical interface has been developed to support the analysis of data obtained from the experiment. To summarize, we developed a microfluidic system that enables advanced 3D cell cultures using hydrogel matrices. We optimized rapid prototyping techniques using laser ablation and 3D printing to create cost-effective, customizable chips. We've ensured biocompatibility and reproducibility, making the system suitable for real-world biomedical applications. Thank you very much for attention. I will now be happy to take any questions.